Okay, welcome to our video about nitrogen analysis. In this video, we'll show you, we'll walk you through all the steps to do nitrogen analysis with our Elemental Analyzer machine. So we'll walk through like sample preparation, milling, how to prepare it for the machine, and then how to run it on the machine.
Okay, so we have the dev moms from different developmental movements of, this is Dickia fragrans, and it's been placed in the oven. We're gonna take some of the sample and we're gonna put it in one of these, how many microliters? Like 10 microliters, I don't know. And then we will put around 3 beads, 3.5 to 4 is usually the best. And then we put in the bead beater for 5 minutes on 1500 RPM. And then see if it's powderized. And if it's not powderized, we put it back in.
Come on.
I'm gonna cut the plant tissues into tiny pieces, and then right here I have my tube labeled. And you also have to make sure you label the cap. I'm gonna put 3.5 beads in here. And then my plant tissue in here. But you wanna weigh the tissue first. Okay. First, make sure you tear the boat. Okay, you would want between 25 to 50 milligrams so that it fills the tube about halfway. Make sure you record the number. Put it in here. Like that, so it's like 2/3 of the way full.
Okay, so once we have the samples in the tubes and they're all labeled on both the cap and the side of the tube, we're gonna load them into the Powerlyzer 24.
You didn't label the other one on the cap.
That's okay, you just should have them all labeled.
Don't forget to label.
These aren't. Okay, so we need to make sure that this is balanced. There's little, the minimum that can be run is 4 samples. There's little triangles on here to show you, to guide you so that it's equally balanced. So we're gonna put 1 on each so that it's even. And then we're gonna take the top and screw it on till it's about like hand tight, but not too tight. And then there's a lock here, which will flip down into the lock position. So once the samples are in, we close the hubcap all the way down. And then here's where we can program how long and how fast it runs. So we're gonna set it to 1500 RPM for the speed, and we're gonna do it for 5 minutes. There we go. Okay, so then we move these arrows to go back up to the top, and it says ready here, and we say start. And then it makes sure that we have the hub lock closed, the plastic, and then we can click the green start button.
And then you open it up, there's a lock, you unlock it, you unscrew it. And right here, I usually look to see if they were powderized okay, and then if they need to be put back in. So that's looking pretty good, can't really tell, but something like that. And usually it'll rub off on the top, so relabeling isn't the worst idea if you need to repowderize.
This is what it should look like powderized, very fine. After you extract as much tissue as you can from the tube, you need to weigh it to see how much that was lost in that powderization process. Make sure to record the number you got. And now I'm gonna transfer it to this tube that has a different endpoint compared to the first one that we used to powderize. This one has a pointier bottom surface, which allows the ball to grind the plant material compared to this one, which is what we're gonna use for storage. This is about the amount that you should expect, and it should stay in here until we analyze it.
Go.
Okay, so we've been putting the beads, the used beads, in a beaker, and then we'll put some water and some lab grade soap, and let it sit and mix it around. But then after that, we've just been putting some in our hand, putting some ethanol on the Chem wipe, and just making sure that the beads are just sanitized. And we've also been using the ethanol to clean all the surfaces before each type, as well as the weigh boats if you're gonna reuse them. We throw this out after we grab the powder. If there's something that's a lot of powders at the sides, you can kind of scoop it, but we're not gonna reuse these. And yeah, just make sure you put ethanol on your tools and wipe them down with a Chem wipe to make sure that they're all dry.
Okay, so now we're in TED Science, and here's the elemental analyzers over here. And so we're going to keep our samples in a desiccator. So we have this big desiccator here. So if we open it up, we're going to keep all our samples uncapped in the desiccator with fresh desiccant in the bottom here. You can see the desiccant underneath. It's mostly blue, but when it starts to turn pink, that means that it's all used up and it needs replacing. And we need to make sure that when we close this, we fully latch both of these all the way down. So this is a little vial of our standard reference material, which is peach leaves, and we keep that and our standard atropine in the desiccator as well at all times in these little glass vials.
So first what you want to do is you just want to make sure that everything is sanitized. I just got the Chem wipe with some ethanol, wiped down the table, wiped down my tools, the little loading block, and also the little packing machine inside there, just to make sure there's no debris. And so we're gonna pick up one of these elemental tin containers. I like to get it right there. Then we're gonna zero. Oop, that was already open. So that makes sense. Gonna close it. All right, and then I just put it in the container. I try to open it up as much as possible so I can get a good amount in there. This is my favorite tool. Everyone has their own. You want to get around 5 milligrams of samples, so I would say it's probably around, this might be too much. And then pick it up, carry it under. And, oops. Here. All right, so after I got the initial open weight, just to kind of know how much we could be losing possibly or adding from air, I'm gonna put it in the little packing machine. You're gonna close the aperture as tight as possible. Sometimes it gets stuck, and then press it down from there. And then gonna weigh and take this weight and record it. So we just want to make sure that the open weight and the closed weight don't differ way too much. Probably about 0.1 is the most that you want it to differ from. And then using this kind of tamping, just press it down so there's no sharp angles and it will slide through the elemental analyzer to be combusted.
Alrighty, so next I'm going to walk through a little bit of the software involved with the elemental analyzer. So we're going to start by opening up this here, and we're going to go to the first. Okay, so now we are in the main console screen. So this is how we're going to control pretty much everything to do with the elemental analyzer. One of the first things to note is how we're going to control the temperature of the machine and when it's in its on, standby, or off settings. So the way that we control this is through this edit button. So we click edit, and then go to Edit Elemental Analyzer Parameters. So I have the machine fully on right now, and so this is how it should look. The left furnace is clicked on, the right furnace is clicked on, and the oven is clicked on, and standby is clicked off. So if I want to put the machine into standby, I'm going to click just the standby button, and the ovens stay all on. And then when all these boxes are checked, I'm going to click send and then OK. And you should hear a little click on the machine. So we put it in standby during the week, so it can sit in standby overnight, but like over the weekend, or if the machine is not going to be used for over like a day or so, then we could need to turn it all the way off. And the way that we do that is we keep standby clicked, but we turn all the ovens off like this. So they should display off on all the ovens, and then we click send and OK. One of the first things we have to do is go into the sample table so that we can update all the samples that we've loaded into the Elemental Analyzer. So we can get to it with this button here. And this takes us into the sample table. So as you can see, for example, we have the sample name, the file name, the type of sample, and then if we're running a standard, the type of standard. So here's a good example of a good run list. We have 5 blanks to start the day, followed by 7 atropine standards. And then we have a QC of the peach leaves, and then 5 samples and another QC. We can extend the amount of samples between these QCs to about 10. We just want to keep an eye that these QCs are not deviating from 10% from the first QC. So once we type all these in, we need to make sure that in the file names stay pretty short, probably no longer than these ones here. And they have the date, like 07/23, like July 23rd. And we need to make sure there's no special characters, no periods, no underscores, just numbers and letters. And then when we're done, we just click OK in the bottom right here. So the software is able to automatically calculate our percentages through this weight column. But the way that we do it is all within Excel, so that we can create our own calibration curve. So there's actually no need to put in the weight here, and we'll just extract the responses from these samples after they're run. So next I'm going to show you how to actually run the samples. So if we check our sample table, we see that I inputted these air blanks that I'm trying to run right now. And so you want to make sure that on this column we have selected the actual sample being acquired to the one we want. And for me, it's this 4th air blank. So I make sure the actual is on the one that I want to do, and then I've labeled the name and the file name and the type, and then I go click OK. So then we're going to make sure that the Elemental Analyzer is displaying ready and normal, and our ovens are at the correct temperatures, like 1000 and like 780. And these 2 lights mean that we're ready to run. So we go back over here, and we have those samples ready, so we go to the run button here, and we just click start sequence of samples. And we don't need to click anything down here unless we want the machine to go straight into standby. We can click this box here, but for now I don't want to do that, so we're just going to click start now. And we're going to hear a little click, and that means it's starting. Okay, so after we've run our samples, we can go into and view our results through this summarize results button next to the sample table. So we click on this, and it takes us into this page, which shows the responses for each of the samples we ran for nitrogen and carbon. So the way we view these is by clicking on the number on the side here, and then we're able to view the chromatogram by clicking up here. So the reason we have to view the chromatogram is that sometimes it doesn't draw the best baseline for these peaks. And so these look pretty good, but the way we can check, especially for our nitrogen peak, is that we can click and drag and make a box around it to really zoom in on it. And we can see that actually the baseline here is not, or the line they drew for the peak is not all the way to the baseline on both sides. So the way that we can fix this is by going to peak and saying move peak start, and we're gonna click right on the baseline here. And then we can click here again, move peak end, and we click on the baseline on the other side. So now that baseline is looking pretty good, And the way that we can now see our new response, which is the area under this peak, is where we go to show and view peak data. And this number here, the peak area, is the number that we're going to extract and put into our Excel sheet. And we can see that that number there is actually has a pretty significant change from the original number that automatically computed for the nitrogen. So it shows how important it is and how much of a difference it makes to redraw those baselines. A couple of things to take note of are that when the instrument is in standby, we're looking for this level to be about like under 2000. And so we want to keep an eye on that, and if it's getting too high, especially if it's like climbing up a lot, like 10,000 more, we want to let Darren know, because that means something might be clogged or wrong with the machine. Another thing to note is that we can click view here and view the sample being acquired, and we can basically look at the chromatogram in live while it's being acquired, so we can see this blank running now. Another thing to note is that while the samples are running, we can edit the sample table, and we can keep adding samples on even while it's still running, and we can check out our past numbers here, but we can't have the sample table or the results table open while the sample ends. The machine won't switch over to the next sample unless the sample table and the results table are closed. So we have about like 4 minutes while the sample is running to edit anything, and then we need to make sure to close that out before the next sample runs. And that's at about 240 seconds, which we can see here.